Hello, I would like to first thank the organizing committee for inviting me to give a talk. I will be talking about resting state functional connectivity. I have no financial interests. We show a task paradigm, a block task paradigm. Here we have control condition followed by finger tapping, which is repeated several times. Again, there are many different methods to analyze, but we get a statistical image, and then we set a threshold, and voxels that pass the threshold is considered corresponding to the task. So if it was finger tapping, we say, okay, this is the sensory motor cortex. I have been interested in the resting state signal, and what is shown here is voxel from outside the brain, which shows that it's much less compared to voxel inside the brain. It's also random in pattern. This is a gray matter voxel. One can see the heart rate, harmonics of the heart rate, the respiration, and low frequency which is present. And this is how typical resting state time series looks like. And so what we did was we analyzed this data during rest in a similar fashion as is done for task activation. And what we found is that that the resting state had significant correlation, and we saw the sensory motor cortex very similar to what was observed during finger tapping. And because Carl Friston had defined functional connectivity as temporal correlation between neurophysiological signal, and because we were observing this at rest, we called this resting state functional connectivity. Although task activation has many advantages, one can think of many cases where a subject will not be able to perform the task, like in infant psychiatric cases, neurological disorders, coma, and so on. So here is a study by Han Zhang, which was published in Neuroimage recently, showing that from one resting state scan, they can map out the motor cortex using both seed-based analysis as well as ICA. They could also identify the visual cortex using seed-based as well as ICA, and the default mode network. Now, this would have been very difficult to perform a task in an infant using task activation. In a famous study, Damien Fair used this resting state data to show the default mode structure, and to show how, as between children and adults, there was different in network properties which could be seen using resting state signal. In another study, Xiong Gong and colleagues used major depressive disorder, MDD, to show differences between MDD and healthy control using resting state. They looked at different graph theoretical parameters, and then found the network structure and tried to correlate with clinical measures. In another study, Helen Zhuo asked this question whether one could show differences not only between healthy control and Alzheimer, but show differences between different kinds of neurodegenerative disorder. So they are showing differences in AD, frontotemporal dementia, and 3 other neurodegenerative disorder, again showing that one can use this to show its differences not just with healthy control but also between the different disease type. One of the criticisms has been, or initial was, if resting state had any neurophysiological basis. And in one of the studies that we had performed, we first did task activation using finger tapping, then this is using resting state. Then the subjects were asked to breathe 5% CO2 for several minutes, and because with breathing CO2 there is a significant increase in blood flow, we saw that there was a significant reduction in these connectivity pattern. And after the subject breathed room air, we could see that these connectivity pattern were re-emerged, suggesting that by using physiology one could reversibly or modulate these fluctuations. In another study, Visakavanimi and colleagues used thiopental to show that with thiopental anesthetic drug, there was an increase in functional connectivity in the visual cortex. However, not all anesthesia are the same. In another study using sevoflurane, Scott Peltier and Xiaopeng Hu showed that in the awake state there was correlation both within and between sensory motor cortex. In the light anesthesia condition there was connectivity pattern within the sensory motor cortex, either in the left side or the right side, but not between left and right. However, with deep anesthesia, the connectivity pattern both within and between was could not be observed. In another famous study, Marcus Reichel and colleagues showed that in macaques with anesthesia, one could see this connectivity pattern, and even with deep anesthesia, one could see the connectivity pattern, although diminished. However, the electrophysiological signal that was obtained had different characteristics with deeper. Task versus rest. So there has been some debate as to what is task activation map and what is resting state connectivity map. While some have suggested that the resting state functional connectivity map is a superset of task activation, others have suggested that while task activation is used for functional localization, a functional connectivity is used to study functional integration. In perhaps the first study where this was explicitly mentioned, in this paper by Jinhu Xiong and Peter Fox, they showed correlation with finger tapping, the first one, the top row being the task, and identified the primary motor, the supplementary motor region. In the bottom row, in the resting state connectivity map, they showed that you could find much more, or I should say, several additional regions associated with the somatosensory cortex, and postulated, as I mentioned earlier, that resting state was a superset of which task activation was only a subset. In another study which was published by Steve Smits and colleague along with Peter Fox, they used the brain map database which had 58,000 coordinates of coactivation corresponding to different tasks. And then they did independent component analysis from 36 subjects, and what they found was that a very good correspondence, let's say for the visual task, the motor task, the default mode, and other region, again suggesting that using the brain map database which had coactivation pattern, you could identify or you could decompose them into a set of networks which was almost identical to the resting state network. So this is the coactivation matrix between 140 regions. This is the correlation matrix from the same 140 regions. And when we look at the correspondence between coactivation and correlation, we find a very good correlation between the two. So we wanted to see if there was some difference between coactivation network and resting state correlation network. So this is showing the graph theoretical measure, the modularity between coactivation, correlation, and a random network. And this is the correlation network showing different regions. This is the coactivation network, and this is the random network. And so using this analysis, what we could find is that for coactivation, the thalamus was more active, while for the resting state, it was much less so. So by doing a comparison between the two, what we see is that for the brain at work, it has higher global efficiency, lower mean clustering coefficient, lower modularity, high degree in the thalamus. The brain at rest, the resting state network had lower global efficiency, higher mean clustering coefficient, higher modularity, and high degree in the left temporal lobe. Anesthesia. While a lot of work has been done to map the brain using resting state, I'll briefly cover about a different application where resting state signal can be used for calibration. The fMRI signal that is obtained is a convolution of the neuronal activity with the underlying hemodynamics, and as a result, the changes could be either due to neural activity or hemodynamic. And to minimize the effect or simply vascular effect or hemodynamic, since we are more interested in the neuronal effect, what has been suggested is to do a breath holding task or a hypercapnic task, from which, again, because with hypercapnia, there is only vascular effect and no neuronal effect, to calibrate this with the bold signal. And so we had the hypothesis that because the underlying hemodynamic function, whether someone is at task or rest, is the same, perhaps one could also use this instead of doing breath holding. And this is showing motor task in the cerebellum and again in the motor cortex. This is calibrating using gray matter volume and breath hold, and one can see a reduction compared to the original task. And by using resting state ALFF parameter and gray matter, we get a very similar calibration effect. And again, the point here is that this could be used instead of breath holding, where in some cases a person may not be able to perform the task. The editor-in-chief of Neuroimage criticized resting state and wrote an editorial saying, does the brain have a baseline, why we should be resisting rest? And the main criticism was that most of what we know about brain imaging or neuroscience has been obtained from task activation studies, so why bother with rest? The second criticism was, how can one correct for artifact when we don't know what the ground truth is? And the third criticism was, at that time at least, most of the fMRI study, both resting state and task activation studies, were done on 20 to 30 subjects, and they said that only if a data was collected, if the result could be replicated using 500 to 1,000 subjects, would, only then would it have any meaning. Since, so that is what we did. So what we did was, we asked every group who had published by that time, which is 2009, on resting state, if they could share their data. Most groups politely declined, but these are the 33 groups who shared their data with us. So this data was collected from many different countries using different scanner, Philips, Siemens, GE, Varian, using different field strength, 1.5 Tesla, 3 Tesla, 4 Tesla, some 7 Tesla data, and even different pulse sequences. And we processed all the data in an identical fashion, and what we found was 20 distinct resting state networks using this data, which, to be honest, we were quite surprised. And because we had more than 1,000 subjects, we could ask very specific questions like, was there difference between male and female, and was there a difference in age? Were there any regions which the correlation went down with age versus were there regions where the correlation went up in age? The other thing, we made all this data publicly available. Another thing that we did, which in hindsight perhaps was a useful thing to do, was we also made public all the scripts that was used to do the pre-processing, as well as generate all the figures. So now there's been a lot of work on looking at the big data. So from 1,000 subjects using data sets like from the Human Connectome project and other large data set, researchers have used much larger data now. So here is a recent study by Anis Abral where he used more than 47,000 subjects. The recent paper in Nature from Damien Fair and Dico Doschenbach uses more than 100,000, and this has now translated into application. Here is a paper by Chao Gan Yan's group where they have used this to classify between AD and healthy control using developing a model where they have used more than 85,000 samples. What is shown here by Young and colleagues is that by leveraging resting state data from over 1,000 MDD sub-patients and 1,000 healthy control, one can find two distinct MDD subtypes based on individual functional connectivity pattern. The first subtype showed severe deviation with positive deviation in the default mode limbic and subcortical areas, while subtype 2 showed moderate but converse deviation pattern. So in the next what I'll do is I'll cover some of the work that we are doing and so most of the EEG data, electrophysiology data that is collected at a very high sampling rate of 100 Hertz or even higher. The fMRI data, on the other hand, uses one data every 2 seconds, so it's very has very low sample. With the multiband sequences available, one can collect data every 600 or so millisecond, so can increase the frequency bandwidth a little bit. So that is what we did to look at the different frequencies as is described in electrophysiology, and what we find is that most of the dominant signal is still below 0.2, and however at a higher frequency, although it's not as robust, but nonetheless we do see some connectivity pattern, and the hope is that with more development and faster scanning method, perhaps better signal can be characterized in the low frequency range. This is just showing differences between healthy control and schizophrenia based on the frequency characteristics. Next, I'll talk about modulations of functional connectivity, and as has been reported by several groups like Katie Chang and others, have shown that the connectivity pattern in the default mode and task positive network varies, changes as a function of time and varies quite a bit. Daniel Handwerker and others have suggested that some of these variation could simply be explained by periodic changes like respiration signal and so on. Elena Allen from Vince Calhoun's group has used like a correlation matrix structure and used principal component to show that there are 7 states, if you will, to look at these data during resting state. So we hypothesize that perhaps there is more widespread activity by a task that are not detected by traditional method. And so we are trying to infer connectivity from task signal, and we use the HCP data using PPI. And so this is the equation for functional connectivity. This is for task activation. And so we are using a regression model with the interaction term. And what we are doing is we are looking at this, right? So this is the 6 block design task from the Human Connectome project, and the stop signal is an event-related task from UCLA group, which is available on OpenNeuro. And after doing this analysis, what we find is that there is widespread both positive and decreased connectivity for the different tasks. For the language task, social cognition task, working memory, and stop signal task. And depending on the task condition, these increased connectivity or decreased connectivity seem to change. So while I have covered about different methods and so on, one of the things I have always worried about is what are the assumptions that we have made. While we can say that, in resting state has a lot of information which the task activation people had assumed was simply random noise. One worry always is that have we made assumptions which are not exactly true? And one of the assumptions that we have made is most of the signal that is present is in the gray matter. However, white matter comprises about 50% of the brain volume, just as much as the gray matter. And recently there has been several papers which has focused on the white matter in task activation study. Here is a review by Jody Garlick and Ryan DRC, where they're showing white matter changes for different tasks. This is another paper by Zhou Ding, where they show that in the white matter tracks, as the task load is changed, these connectivity pattern in the white matter also is modulated, suggesting again that white matter does play a role. In another study by Michael Pierre, which was published in Journal of Neuroscience, they used the white matter signal and used k-mean clustering to show that the white matter, just like the gray matter, had distinct networks. Again, what is shown here is the reliability of this white matter functional network. For the different networks, we may show the scatter plot between stationary functional connectivity and dynamic functional connectivity for gray matter, white matter, and gray matter, white matter networks. And then here we show similarly the reliability of these networks. So what we did was we focused on corpus callosum, one of the largest white matter structure, and we used the human connectome data. And what can be seen here is we are showing specific regions of the corpus callosum which correlates to the specific white matter network. Right, so this is one, the second, method, and so on. And again, this one, because we had used the HCP data, and there was a second scan, we used it, and then they showed that these results are fairly reliable. And because the Human Connectome project also had the DTI data, and the corpus callosum has been widely described, using DTI, we mapped the corpus callosum into the specific subregions, and this is showing the DTI region corresponding to the resting state region for each of the subregions of the corpus callosum. And the overlap is the one shown in red. While it's not a perfect overlap, nonetheless we see several regions, or I should say several subregions of the corpus callosum which match both with resting state functional connectivity as well as DTI. In another study by Li Jiao and Liao Wei, they looked at the graph theoretical properties of the white matter signal and compared it to the gray matter. And when they analyzed the signal characteristics, what they found was that there were differences in the structure, the graph theoretical structure, again suggesting that the white matter correlation that we observe is simply not due to a leakage, if you will, from the gray matter signal. So one question has been that could this have any electrophysiological basis? Just recently, Dr. Chui published a paper in Nature Neuroscience where he used this. He used EEG data and then showed a good correlation between bold functional connectivity and EEG functional connectivity, and again suggesting that perhaps there is electrophysiological basis to these connectivity patterns. Again showing differences between in the white matter in the AD subjects. Figure on the left shows decreased clustering coefficient and local efficiency in gray matter or white matter region suggests the disrupted information processing efficiency in AD. On the right side, what is shown is the significant correlation between FA and nodal topological metrics, which may provide new evidence on the association between structure and function in white matter, suggesting a structural basis for bold fMRI signal in the white matter. And then trying to link it to the different gene profile. So again, these are work in progress, which I'm happy to talk in more detail if someone has questions. There has been a number of clinical applications which has emerged. I'm just listed a few. The last thing I wanted to cover is naturalistic stimulus. And there has been a lot of interest in recent years about movie watching. Although movie watching as a paradigm was suggested in 2004 by Yuri Hassan, which was published in Science, and then what he had done was he had showed that while using this paradigm, you could find activation corresponding to fusiform gyrus. The other interesting finding in that study was because all the subjects were watching the same movie, the time series from one region could be taken and correlated with a different subject to show correlation. So what we wanted to specifically do is that because there's many tools now in resting state network, we wanted to ask whether dynamic or stationary connectivity is a better measure, and what can it compare to. So for this one we used three different data sets. The partly cloud data, data from the Healthy Brain Network serial scanning. So this is the dynamic connectivity for three different movies: Wall-E, The Matrix, and A Few Good Men. And in this, what is shown is that for the different ICA components, the interaction is fairly different for each of the movie. What is shown here is the stationary connectivity and age effects and sex effects for the two different movies. For the dynamic connectivity for the same two movies, we can see both the age and sex effects, and in this case it seems more clear compared to the stationary connectivity. So the dynamic connectivity more sensitive to states, the movie content, then stationary connectivity. The dynamic connectivity is more sensitive to age effects and sex effects. The dynamic connectivity may be more functionally relevant. However, limitation is the difficulty to directly link dynamic connectivity to movie content. Of course, another question is that if the connectivity pattern is in a specific way while watching movie, and the resting state connectivity is not, then what does it imply? There's a lot of work that still needs to be done in terms of modulation to better understand how can we measure these functional connectivity, either using physiological challenges like breath holding hypercapnia, or using anesthesia or TMS, TDCS. In terms of methodological development, in addition to perhaps developing pulse sequences which can collect specific kinds of data which could be sensitive to more flow or ball weighted, or collect data at a faster rate. Analysis method could perhaps be used to better describe multimodal information, combining white matter analysis and gray matter analysis in a single pipeline think there's a lot of work which is currently being done on validation methods. Last but not the least, I would like to thank all my students and collaborators. Thank you.